Hello dear students, this is Dr. Adwet and welcome to lecture number 11 of Cell, the Unit of Life. As far as this chapter is concerned, let's have a quick recap. In lecture number 1, we saw Introduction to Cell. In lecture number 2, we spoke about history of microscopy and the cell theory. In lecture number 3, we spoke about parts of a cell, basics of genetics, characteristics of a cell. In lecture number 4, we studied the nucleus. In lecture number 5, we saw plastids. In lecture number 6, we spoke about mitochondria. In lecture number 7, we saw ribosomes and endoplasmic reticulum. In lecture number 8, we saw Golgi bodies, lysosomes and vacuoles. In lecture number 9, we discussed the cytoskeleton. And in the last lecture, dear students, we have seen the plasma membrane. So all these things that you see here are the respective lecture numbers and what we did in that particular lecture. Also, please remember that all of this has been in regards to a eukaryotic cell. So, as far as a eukaryotic cell is concerned, we know that some eukaryotic cells like plants and fungi are going to have a cell wall, and inside the cell wall is the living material called as protoplasm. Protoplasm is further classified into the nucleus, the cytoplasm with the cell organelles and the plasma membrane. We finished the nucleus long back. We have finished all these organelles which include plastids, mitochondria, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles and cytoskeleton. We have also finished off everything related to the plasma membrane. So the nucleus, the organelles and the plasma membrane of a eukaryotic cell are done. Which means that the last thing about the eukaryotic cell that we need to finish is the cell wall. And that, dear students, is going to be today's lecture. Okay, so let's start today's lecture, which of course, as I just told you, is going to be on the cell wall. Now, as far as the cell wall is concerned, this is a diagram that you already know, and this is what we had done in lecture number 3. So you can see that what I have drawn here is a cell with a nucleus, and this cell which you see here is going to have a plasma membrane or a protoplasmic membrane or a cell membrane. Within it, it is going to have the cytoplasm, and inside that it is going to have the nucleus with the nucleoplasm. So the nucleus, the cytoplasm and the plasma membrane, all of this is living, and it is also going to be called as the protoplasm. In a few eukaryotic cells like plant cell, fungus, some protists, what we find is that surrounding this protoplasm is a non-living thing which is going to be called as the cell wall. And that, dear students, as I said earlier, is the discussion for today's lecture. Now, as far as the cell wall is concerned, you all know that it was first observed by Robert Hooke. Robert Hooke was the first person to observe it, the same person who drew that entire book that we spoke about called Micrographia. After that, what we need to know about the cell wall is that it is the outer, rigid and semi-transparent part in a few eukaryotic cells, not found in all eukaryotic cells. And as we discussed it in the third lecture itself when we were seeing this is usually going to be found in the case of a plant cell. This is usually going to be found in the case of a plant cell. Plus, you already know that it is non-living. This particular cell wall, this particular brown colored cell wall that you see is not living, because in the third lecture itself I explained to you कि अगर ये living है तो उसको protect करने के लिए उसके बाहर और कुछ living चाहिए, non-living चाहिए होगा. अगर वो भी living होगा तो उसके भी बाहर कुछ तो non-living की ज़रूरत पड़ेगी. So of course cell wall is going to be non-living, and if it is non-living, the material which makes up the cell wall has to be made by something which is living. So who is going to secrete all the material of the cell wall? Yes, that is going to be the protoplasm, or it is going to be the cytoplasm of the cell surrounding which we have this cell wall. So the non-living secretion of the protoplasm or cytoplasm is called as the cell wall. Also, it is seen in plants, fungus, bacteria and certain protists. So in eukaryotic cells like plants and fungus, it is seen. It is also seen in prokaryotic cells like bacteria. In bacteria or prokaryotic cell, it is slightly different. Today, we are only going to be talking about eukaryotic cell. Right. Lastly, it varies from 0.1 to several microns or micrometers in its thickness. So depending on the age of the plant, age of the cell, condition of the cell, there are various factors depending on which the thickness of the cell wall will be decided. So it can be very thin, about 0.1 microns, and it can be even several or a few hundred microns. So that is the introduction that you need to know about the cell wall. Now here I would also like to mention a few things that we had done in lecture number 3, which was that a plant cell will have this structure which is going to be rigid, will not show too much movement, which is going to be thick, which is going to be tough, which is going to surround the cell. And at that time I told you that the part inside, protoplasm, can move and change its shape. But when you see the outer part, which is this rigid structure called as the cell wall, that part is not going to move. So a plant cell is not going to change its shape, whereas an animal cell which does not have this cell wall may be able to change its shape. So animal cells usually can change their shape, plant cells cannot change their shape. Also, because of this cell wall, the plant cell is going to be slightly larger as compared to a few corresponding animal cells. So these two things will be important later on in today's lecture, that plant cells are usually larger as compared to an animal cell, and plant cells usually do not change their shape, whereas an animal cell can change its shape. And of course, plant cells are going to have cell wall and animal cells do not have cell walls. Right, let's move ahead. If we were to take a section of this particular cell, which you can see in the box up there. So we've taken a part of this cell and I'm going to enlarge it. So now that I have enlarged it, you can see that the innermost part is going to be the cytoplasm. The black line surrounding it is the plasma membrane, and outside of that you are going to see the cell wall. The cell wall which is dead, not living, but it is made by material which is secreted by the protoplasm. So यहाँ पे जो साइटोप्लाज्म या प्रोटोप्लाज्म है वही ये सेल वॉल को बनाने वाला है, जो सेल वॉल तुमको यहाँ पे दिख रही है. Now, the thing is that by looking at such diagrams, you may come to the conclusion that the cell wall is a very wall-like structure. दीवार जैसा, literally bricks से बनी हुई दीवार जैसा ये structure है जो अंदर की चीज को protect करेगी, बाहर से अंदर कुछ भी नहीं आएगा, एकदम like a wall it is standing. But that, dear students, is not actually the case. The cell wall is not a wall like this which is completely fortified and which cannot be penetrated. In fact, a real illustration of a cell wall, a plant cell wall, would look somewhat like this. So what you can see here, is going to be the plasma membrane. Somewhere here will be all the cytoplasm, and everything above this plasma membrane, is the cell wall that we are talking about. So all of these things which you see here is going to be parts of the cell wall. All the things that you see there are going to be parts of the cell wall. Notice how it is not brick by brick and completely fortified. In fact, you will notice that this entire area has this yellowish colored matrix. Have a look at this yellowish colored matrix that you have here. And in that matrix, there are these dense fibrous rods. Can you see these rods or these cylinders here, which are actually bundles of fibers called cellulose? And I will explain that in more detail in a minute. So what you have here are these cellulose bundles in this yellowish diffuse matrix, which is mostly water and some other chemicals called as hemicellulose and pectin. So there is a matrix here which you can see of hemicellulose and pectin. In fact, if you were to notice these structures here, these structures which are in blue, that is hemicellulose. The cylindrical structures which you see are going to be a chemical called as cellulose. And above this you see a thin gelatinous membrane. Now that thin gelatinous membrane you must be seeing in the diagram is called middle lamella. All of this here you can see is called primary cell wall. ये सारी चीजें हम लोग आज डिटेल में करने वाले हैं. अभी उस पर ध्यान मत दो. अभी के लिए सिर्फ इतना समझो कि ये प्लाज्मा मेम्ब्रेन है, and outside the plasma membrane, this whole structure that you see here is the cell wall. And as far as the cell wall is concerned, it is going to have a diffuse matrix in which there are these cylindrical structures which are going to be called as cellulose. The diffuse matrix will be made up of compounds which are pectin compounds, hemicellulose, mostly around 60% is going to be water, and all these things we are going to discuss now in detail. But this picture I am showing you right at the onset so that you understand that the cell wall is not completely impermeable and fortified जिससे अंदर बाहर कुछ जाएगा ही नहीं. It is going to look somewhat like this as you see in this image. Correct? So let's move ahead. So the next thing that we have to now discuss is that from this diagram itself, it should be clear that the spaces in between the fibers will allow movement of molecules which are small enough to pass through those spaces according to their gradient, जो हम लोगों ने last time ही देखा था. So anything which is on this side or on this side can freely move from here provided it is going to be a small molecule. So remember that this cell wall that we are talking about, the primary cell wall, is going to be freely movable for small little molecules. Now let's proceed with the cell wall, and as far as the cell wall is concerned, let's first talk about its chemistry. Now to discuss the chemistry or what are all the molecules involved in making the cell wall, let's first divide them according to which region they are present and what do they do. So you are going to have the matrix, which I explained to you, in which you are going to have the fibers, and there may be some other materials other than fibers also which may be present in the matrix. So we simply classify that as other components. So you have the matrix, you have the fibers, and you are going to have the other components. Now once again, I would like to bring this diagram to your notice, where you can see that all this diffuse yellow bit that you see here in between is the matrix. These blue fibers are hemicellulose, and these thick bundles or cylinders are made up of cellulose. And all these things we are now going to discuss in detail, right? Let's proceed. So as far as the matrix is concerned, it is about 60% water. It is going to have hemicellulose, which is about 10 to 15%. It will have pectic compounds or pectin compounds, which is going to be about 2 to 8%. It will have a few proteins, 1 to 2%, and it will have lipids, which is about 0.5 to 1%. So this is that diffuse substance within which you are going to have the fibers. So these microfibers or microfibrils will be either of cellulose, which will be found in the case of higher plants. If we are talking about fungus, it will either be fungal cellulose or it will be chitin, and in the case of bacteria, they will be mucopolymers. So basically the matrix will be more or less the same in plants also, in fungus also, in bacteria also, but the fibers which are present in the matrix, which are going to provide the strength, which are going to protect the cell which is within the cell wall, those fibers are different in the case of a plant, in the case of a fungus, in the case of bacteria. Even in different plants, you will have different kinds of fibers. So those are going to be the fibers which are found within the matrix. A few other compounds which may also be found in the matrix are lignin, which provides strength. You have suberin and you have cutin, which have to do with water loss. And finally, you are going to have silica compounds, which if deposited in the cell wall will provide extra stiffness and rigidity. So I am sure that you may have questions in your mind right now regarding कि भाई ये cellulose क्या है, ये fibrils के बारे में आप क्या बोल रहे हैं, hemicellulose, pectin क्या होता है, what is lignin. So all these things we are going to discuss first, and only then we will proceed with the structure of the cell wall. So let's get this little bit of chemistry out of the way so that we can move on to the structure. Okay. Now, what you see on the screen right now is going to be this single red structure, and it can be a type of carbohydrate, it can be an amino acid, it can be something which is a single piece which I can call as a unit. If we take many such single units and if we join them together with some kind of bonding, this structure that we get, which is made up of such many units, is going to be called as a polymer. Now, once you have a polymer, the unit can no longer be called as the unit, because this polymer has many units. So each unit within the polymer will be called as a subunit. And if this is a polymer, if this what you see here is a polymer, then its subunit will be called as a monomer. So many monomers will form a polymer. Many monomers will form a polymer. Simple. So you can have amino acids. So if the monomer is an amino acid and if you join it together, you will get something called as a protein or a polypeptide. If the monomer is going to be some sugar, you will get something called as polysaccharide. So monomers are subunits which join together with bonds to form the polymers. Now, especially in this polymer, what you will notice is that all the subunits are same. They are of the red type, which I have illustrated. That is why this polymer will be called as homopolymer. Homo meaning same. Contrast this homopolymer with this other polymer. So you can see that the structure above is a polymer made up of subunits called monomer. The structure shown here below it is also going to be a polymer made up of subunits. The difference is that in the example above, all the subunits are of the same type, whereas here all the subunits are not of the same type. They are different. And I told you in the first lecture itself that the word for different is hetero. So this structure is also a polymer, but it will be a heteropolymer. So you have homopolymers and heteropolymers. If the subunit monomers of a polymer are same, you can call it as a homopolymer. If the subunit monomers of the polymer are different, then you will call it as a heteropolymer. So I hope that this structure and this illustration is now clear to you. Right. So with this in mind, we can start the next topic, which is cellulose versus hemicellulose. What is the main difference? Because the words seem to be the same. दोनों में cellulose आ रहा है. So what is the difference? Now, both of them are polymers and both of them are hydrophilic. Hydrophilic, so I hope you understand that that means that they are water loving, which means they will have polar units. They are going to have some parts which are going to be polar and some parts which are going to be having charge, and that is why they are going to be hydrophilic. So both of these compounds are hydrophilic, they will be attracting water, and they are both polymers. So what is the difference? What is the difference between these two? Right. So cellulose is a polymer which is derived exclusively from glucose, which means that the only monomer is glucose. So it is a homopolymer. All subunits are same, glucose. So cellulose is a polymer of glucose, made up of monomers called glucose. At the same time, hemicellulose comprises of diverse sugars. So it's a heteropolymer which is made up of multiple sugars. It's not necessary that the same sugar will be repeating in the polymer of hemicellulose. So sugar monomers in hemicelluloses can include 5 carbon sugars like xylose mainly. So most of the time the sugar which is repeating as a monomer in hemicellulose is going to be xylose. Sometimes there are 5 carbon sugars like arabinose, and there may also be 6 carbon sugars like glucose, mannose and galactose. So you can see that in the case of cellulose, the only monomer sugar subunit is going to be glucose, whereas in the case of hemicellulose, you can have various types of sugars which may combine together to form a polymer, and that will be called as the hemicellulose. Also, cellulose is very long chain polymer. So many glucose molecules will come together and form the polymer of cellulose. Almost 7,000 to 15,000 glucose units can come together. In the case of hemicellulose, it is shorter with chains ranging from 500 to 3,000 sugar units. At the same time, cellulose is unbranched. It does not show any branches. But hemicellulose, depending on which type it is going to show branching. So hemicellulose is a branched polymer, cellulose is an unbranched polymer. Cellulose is crystalline, it is strong, and it is difficult to break the bonds between cellulose, the glucose-glucose bonds between cellulose in the cellulose molecule. At the same time, because hemicellulose is going to be much shorter and its structure is going to be quite random, It is not going to be in straight chains, but it is going to have branches, and where those branches are, how those branches are, will depend upon what kind of hemicellulose it is and which are the sugar subunits which is making the hemicellulose. So it has very little structural strength. So that is why hemicellulose is a part of the matrix, and its main job is to hold the cellulose fibers together. And cellulose is going to be the main structure which is going to form the cell wall. So I hope that this distinction between cellulose and hemicellulose is now clear with everybody. Okay. So those of you who don't know, what you are seeing here is a cotton ball, a cotton flower, and as you all know, so right now I am also wearing a cotton shirt. So these fibers are basically cellulose fibers. Cotton fibers represent the purest natural form of cellulose, containing more than 90% of the polysaccharide or the polymer. At the same time, all of you know that if right now during the rainy season, if you were to go out and if you were to wear this, you will get completely wet, because this thing is going to absorb water, because cellulose is going to be hydrophilic. So I hope that all these things are now making sense to you. Right. Just to drill this completely into your head, have a look at this diagram, have a look at this illustration, and you will notice that this structure here is a glucose molecule. So this is one glucose molecule, this is the next one, this is the next one. So what you have here is a single strand or a single polymer of cellulose. This is a single polymer of cellulose, right? Now you can see that here there are 3 strands of cellulose polymer. This is number 1, this is number 2, and this is number 3. And I just told you that they are going to be polar, and if they are going to be polar, that means if they come close by, some negative part of one molecule will attract the positive part of the other molecule, and it will form weak hydrogen bonds. So notice how you have these weak hydrogen bonds which are formed between this. So these 3 individual strands will now come together and will form a very tight bundle. Why will it form a very tight bundle? Because it is going to be polar, and because it is polar, it is going to form these hydrogen bonds and come together and form a thick bundle. So this thick bundle, which is made up of many such strands, is going to be called as a microfibril, as you can see here. Many such microfibrils will form a bigger bundle, which is called as a macrofibril. And this, dear students, this macrofibril is what I showed you in the previous diagram. So the actual diagram of the cell wall, where we saw the matrix and we saw the bundles of cellulose, these are the bundles of cellulose that we are talking about. That cellulose strands come together to form something called microfibril, and many microfibrils come together to form a macrofibril, and the macrofibril of the cellulose molecules are going to be in the cell wall. Here. So what you see in this image here, is not just one polymer of cellulose. It is many strands of cellulose forming microfibril, and many microfibrils joining together to form macrofibrils. And these macrofibrils are what you see here. As I said, they are extremely tough, they do not break down, and because of this bonding, they become like a bundle which is even more tougher. And that, dear students, is what you find as cellulose in the cell wall. Okay, moving on. Now, along with this, you will notice that the matrix has water, that you know, hemicellulose, that also now you know. It also has pectic substances. Now, these pectic substances or these pectin compounds, let's just do a little bit of their chemistry in brief. Pectin is consisting of a complex set of polysaccharides contained in the primary and middle lamella layers of the cell walls of terrestrial plants. So this entire line you will understand at the end of today's lecture, but right now it's sufficient to know that pectin is going to be found in the cell wall. So what is pectin? It's a polysaccharide, and its main component is galacturonic acid, and galacturonic acid is a sugar acid which is derived from galactose. So the acid which you can get from a sugar called galactose is going to be called galacturonic acid, and that is one of the main components of this group of compounds called pectins. So pectins are basically polysaccharides, sugars, polysaccharides. Now, pectin helps to cement the walls of adjacent cells together. So think of pectin as that sugary jelly which is going to chipka two cells together. So it is going to take two cells and it is going to literally cement them together. So it is a sticky jelly-like substance. In fact, because of its ability to form a thick gel-like solution, pectin is used commercially in the preparation of jellies and jams. So you all know of sweets which are available in this form, and the compound which helps them become like this, become in this state of matter, jelly-like, that compound is usually going to be pectin, some sort of a pectin compound. So pectin compound is something which is going to be rich in galacturonic acid, which comes from the sugar, the polysaccharide galactose. So as far as you are concerned, you just need to remember right now that pectin is something which is used to cement cells together because it has properties like this. So that is what you need to know about pectin. Now, the other things which were in the last column. So we spoke about everything in the matrix. We saw all the fibers, the cellulose fibers, the chitin or the fungal cellulose fibers, and the mucopolymers which are found in bacteria are also having more or less the same structure. So now we come to the last column, which had words like lignin, which had words like suberin, cutin, and silica compounds. So first let's talk about lignin. Lignin again is a class of complex organic polymers that forms the key structural materials in the support tissue of vascular plants and some algae. Again, as we move with the syllabus, this line will become more clear, but abhi ke liye you need to know lignin provides support. Lignin provides support. Now how does it basically provide support? Lignin fills the spaces in the cellulose, hemicellulose and pectin components, thus providing mechanical strength to the cell wall. So about a couple of minutes back, I showed you that diagram again of the cell wall with the cylinders of cellulose and hemicellulose, and surrounding it was the matrix. Now imagine a tough material which is difficult to break down called as lignin fills in that space. So if it fills in that space, and if lignin is going to be hydrophobic, what do you think is going to happen? The entire cell wall will go from being permeable, being hydrophilic, and being slightly flexible. Now from these properties, it will go to being inflexible, being highly thickened, and instead of being hydrophilic, now it is going to have water repelling or hydrophobic properties. So lignin fills the spaces in the cellulose, hemicellulose, pectin components, thus providing mechanical strength and support. The polysaccharide components of the plant cell walls are highly hydrophilic. So what are the polysaccharide components? The cellulose and the hemicellulose. So the polysaccharide components of the plant cell walls are highly hydrophilic and thus permeable to water, whereas lignin is more hydrophobic. The cross-linking of polysaccharides by lignin makes the cell wall less permeable to water. So lignin will ensure that all these polymers link to each other and lignin will fill up the spaces in between. Thus, when you lignify a cell wall, the cell wall becomes impermeable to water. And when the cell wall becomes impermeable to water, no water or very little water can enter the cell. So technically the cell dies. So a lignified cell is a cell which is going to die. But a lignified cell is going to have a very thick cell wall which will provide tremendous support to the plant. Once again, all these things are going to come in much more detail in the future chapters. But आज के लिए I hope कि lignin का तुमको थोड़ा बहुत idea आया कि it is a tough polysaccharide, it is a complex polysaccharide, it is difficult to break down, it is going to be hydrophobic, and if it gets deposited in the cell wall is able to provide tremendous support but loses all flexibility. Right. And lastly, lignins are also particularly important in the formation of cell walls because they lend rigidity and do not rot easily. Meaning that lignified cells do not lose their shape at all. Of course, lignified cells are dead, like how I explained to you, but they will maintain their shape and the lignin will not break easily. So these are all the things which you need to know about lignin, which is found in the cell wall. Okay, the other two things that were found in the cell wall which were not a part of the matrix, which were not a part of the polymers, was going to be suberin and cutin. So cutin and suberin are cell wall associated glycerolipids. So these are lipids. Now the minute I say lipids, it should be clear that they are hydrophobic. They are going to be non-polar. So cutin and suberin are cell wall associated glycerolipids, polymers that are specific to plants. Cutin forms the framework of the cuticle sealing the aerial epidermis. Now, I don't know if you have done this in school or no, but leaves on their upper surface, called as the upper epidermis, have a waxy substance so that there is no water loss from the upper surface. That waxy substance is called as cuticle, and the compound which is found in cuticle which gives it that property is cutin. So cutin forms the framework of the cuticle sealing the aerial epidermis. Suberin is a waterproofing waxy substance found in higher plants. So suberin is also something similar, but suberin is not found on leaves. Suberin is usually found on the outer part of the bark, and it is going to be found in the roots. So this is what you need to know about lignin, which provides mechanical support and strength to the plant. As far as cutin is concerned, it will make the surface impermeable to water and prevent the loss of water for the plant. The plant will not lose water from its leaves. And as far as suberin is concerned, it is found in the outermost part of the bark, and it is found inside the roots, and what it does there is going to be done in detail in the coming lectures. So आज के लिए I hope that this chart is now clear to everybody as to which things are found where within the cell wall and what are the specific unique properties of each of these components that you see here as far as a cell wall is concerned. So all of this was relating to the chemistry of the cell wall. Now I am going to actually teach you the structure of the cell wall, and if you understood this, then the remaining part of the lecture is going to be extremely easy. So let's start. Let's say that these are two newly formed plant cells. अभी सेल डिवीजन हुआ है और ये दो प्लांट सेल्स यहाँ पर आए हैं जो आपको यहाँ पर दिख रहे हैं, Now these plant cells, the minute they are formed, we want them to stick together. We don't want them to move away, we want them to remain close by. So we need some material which is going to ensure that these plant cells remain stuck to each other. So in between these two newly formed plant cells, you will get this membrane which will be secreted by the protoplasm of these cells. Now this thing which you see here is a thin and amorphous layer. So it is thin and does not have a proper definite shape. Thoda upar niche ho sakta hai. Here I have shown it as a straight line, but it can be slightly up or down. This, dear students, is going to be containing calcium and magnesium pectates. Pectin salts. The same pectate or pectin that we were talking about which cements the neighboring cells. This is called as the middle lamella. So I hope you have understood the concept of what is a middle lamella. So it is in between these two cells, and because it is a sheet, it will be called as middle lamella. Please remember that the first component of the cell wall to be made when a plant cell is divided is going to be the middle lamella. So अब तक cell wall वो cellulose, hemicellulose सब जो बोला वो तो अब तक आया ही नहीं है, पर अब तक ये दोनों cells के बीच में ये जो चीज़ आई है पतली सी amorphous, that is going to be called as the middle lamella. Please remember, the middle lamella is the first component of the cell wall to be made when the cell wall formation starts. Clear? Okay, so middle lamella. Now, next what is going to happen? The plant cell, as in this cell and this cell, will now from their protoplasm, this living material, they will start making cellulose, they will start making hemicellulose, pectin, and they will start pushing all of that towards the outer part of the cell ताकि cell के बाहर क्या non-living चीज बना सके? Cell wall. So now this is what is going to be produced. Can everybody see that? So that dark brown color thing is going to be thin and elastic. It will be able to move. It is going to be inner to the middle lamella. So as you can see in this diagram, वो येलो लाइन के अंदर की तरफ है, येलो लाइन के अंदर की तरफ है, मिडल लैमेला के अंदर की तरफ है, towards the cell. So it is inner to the middle lamella. Its matrix is made up of water, pectin and glycoproteins. The microfilaments which are embedded within the matrix are going to be cellulose, and it grows by intussusception. Now what is intussusception? Intussusception meaning growth from within. So if this thin and elastic brown color ki membrane for some reason gets pulled or stretched, then there will be gaps in between those cellulose fibers that we were talking about. And there in those gaps, there will be more deposition of cellulose. मतलब कि ये मेम्ब्रेन को खिंचाव तनाव से उनके बीच के जो सेल्यूलोज फाइबर्स के बीच के गैप्स है उनको थोड़ा बड़ा किया जाएगा और वही गैप्स में और सेल्यूलोज डिपॉजिट किया जाएगा. So since this membrane is growing from within by adding material within the membrane, it is going to be called as intussusception. So intussusception basically means growth from within. So this brown thing, dear students, that you see here, which is thin and elastic, inner to the middle lamella, matrix is made up of water, proteins, glycoproteins. The fibers which are going to be mostly present are going to be cellulose, and which also grows by intussusception. This brown membrane, dear students, is called as the primary cell wall. Is called as the primary cell wall. So as long as a plant cell has a primary cell wall, it can continue to grow. It can continue to grow because the primary cell wall is flexible. So as the inner cell, जो अंदर है, brown चीज के अंदर है, as that cell continues to grow, it can push the primary cell wall, and the primary cell wall being thin and elastic can be stretched. As it stretches, there will be gaps which are going to be produced जहाँ पे और cellulose डाल के उसको भी थोड़ा grow कर सकते हैं. So primary cell wall, please remember, is capable of growth. Now, what happens when the cell has reached its maximum size and now the cell basically wants to provide support? This cell is now willing to die so that it can provide support to the plant. Now why does the cell need to die to provide good support? Because on this cell wall that you see here, the dark brown colored cell wall, you can keep depositing cellulose and make it thicker and thicker and thicker. But if you really want to make it extremely sturdy, you will have to deposit lignin. And what happens when you deposit lignin? It becomes very tough, it does not rot, but it becomes hydrophobic. So water cannot come into the protoplasm. So in certain cells, not all cells, what can happen is more deposition of cell wall material on the primary cell wall. So now please remember that the light brown thing that you see here is not inside the primary cell wall. It is not dark brown के अंदर. So अगर वो dark brown वाली जो line यहाँ पे दिख रही है primary cell wall, उसको खींचा और उसके अंदर cellulose डाला और उसको ही thick बनाते गए तो it is the primary cell wall. But अब तुम वो dark brown primary cell wall पे अंदर से can you see कि ये light brown deposition कर रहे हो? So this is not intussusception. You are not putting it within. You are depositing material on the primary cell wall. You are depositing material on the primary cell wall. And of course, this is inner to the primary cell wall. It's also inner to the primary cell wall. So this, dear students, is going to be called as the secondary cell wall. This is called as the secondary cell wall. So it is seen not in all cells, but in some mature cells in which growth has stopped. They no longer need to grow. They no longer even need to be alive. So in some, only extra cellulose is deposited where they will continue to remain alive, but the cell wall will be very thick. And in some, the cell wall will be made extremely thick by depositing lignin, and in that case the cell will die. So some examples of these cells are going to be tracheids, vessels, collenchyma. All of this is in detail in some other chapter. But today I just want you to know that if you make the primary cell wall more thicker by depositing material from within, because who makes all this material? The protoplasm. Where is the protoplasm? Inner to the primary cell wall. So now instead of growing it from within, primary cell wall के ऊपर अंदर से और material deposit किया जाएगा. So this secondary cell wall is inner to the primary cell wall, and it is going to have more deposition of cellulose, and it will also have more deposition of lignin. If the need be, if it is in a few particular areas in the plant, you can also have suberin, you can also have cutin. Now, this goes by accretion. This secondary cell wall grows by accretion. What is accretion? To deposit upon. So intussusception was growth from within, whereas accretion is growth on top of something. So now you are making layers and layers and layers, new layers inner to the primary cell wall. New layers inner to the primary cell wall. Okay. Lastly, the secondary cell wall usually has multiple layers. The secondary cell wall usually has multiple layers. Meaning के secondary cell wall पे जो सबसे बाहर की तरफ लेयर डाला है, मतलब प्राइमरी सेल वॉल के ठीक नीचे जो लेयर है, somewhere here, somewhere here, somewhere here, somewhere here on this side, that is called S1. फिर उसके नीचे ये वाला जो बनेगा, ये वाला जो बनेगा will be S2, और जो एकदम ही अंदर है यहाँ पे will be called as S3. So usually the secondary cell wall has three distinct layers, S1, S2 and S3. S standing for secondary. Now, in some cases, S3 is also called as the tertiary cell wall. S3 can also be called as the tertiary cell wall. So just to clarify, ये जो तुमको इधर दिख रहा है, ये प्राइमरी सेल वॉल है, ये सेकेंडरी सेल वॉल है लाइट ब्राउन का, उसका सबसे बाहर वाला S1 है, बीच वाला S2 है और अंदर वाला S3 है. जो सबसे अंदर वाला S3 है is sometimes also called as the tertiary cell wall. Is also called as the tertiary cell wall. Now it is called as the tertiary cell wall because जो S1 S2 है, यहाँ का जो लेयर है S1 और यहाँ का जो लेयर है S2, उससे सबसे अंदर वाला लेयर थोड़ा अलग है, मॉर्फोलॉजी शेप में भी और केमिस्ट्री में भी. The S3 layer has more of compounds called xylins, XYLANS, and because of that it is sometimes also called as the tertiary cell wall. So this, dear students, completely finishes off the structure of the cell wall. You know the primary cell wall, you know the secondary cell wall, and you also know the middle lamella. So as you go from the protoplasm of one cell towards the protoplasm of the other cell, what are you going to find? You will find plasma membrane of the cell, secondary cell wall, primary cell wall, then the middle lamellae, then the primary cell wall of the second cell, the secondary cell wall of the second cell, the plasma membrane of the second cell, and then you will find the protoplasm of the second cell or cytoplasm of the second cell. So I hope this clarifies everything about middle lamella, the primary cell wall and the secondary cell wall. So as far as the cell wall is concerned, you are going to have the middle lamella, you will have the primary cell wall and the secondary cell wall. So now I'm just going to repeat the things that we saw in the last 10 minutes. The middle lamella is thin, it is amorphous, it is the first layer to be deposited during cell division. It contains calcium and magnesium pectates. Main function is to cement the neighboring plant cells. The primary cell wall is thin but it is elastic, capable of growth and movement. It is inner to the middle lamella, it is capable of growth, grows by intussusception, generally diminishes as the cell matures. So as the cell matures, the primary cell wall will become thinner and the secondary cell wall will start increasing in size by accretion. Has matrix comprising of water, pectin, glycoproteins, and has microfilaments of cellulose, fungal cellulose, chitin and peptidoglycans or mucopolymers, which are seen in the case of bacterial cell. The secondary cell wall is seen in some mature cells in whom growth has stopped. For example, tracheids, vessels, collenchyma. It is inner to the primary cell wall. It grows by accretion. Usually has three distinct layers, S1, S2, S3. The innermost layer is sometimes also called as the tertiary cell layer. So now let me show you an electron microscope picture of the cell wall. You will notice that this is cell 1, this is cell 2, and in between these two cells, you are seeing the structure which I just discussed. That this here is the plasma membrane of the second cell. This is going to be the secondary cell wall. Here is going to be the primary cell wall which will be very thin. ये जो तुमको इधर दिख रहा है वो मिडल लैमेला है, ये जो यहाँ पे दिख रहा है वो प्राइमरी सेल वॉल है, ये जो तुमको इधर दिख रहा है वो सेकेंडरी सेल वॉल है और यहाँ पे साइटोप्लाज्म ऑफ सेल 1 है. So you will notice that this is the middle lamella, this is the primary cell wall, and there are 3 layers of the secondary wall. Now those three layers are usually S1, S2 and S3. जो सबसे यहाँ वाला है will be S1, जो इधर वाला है will be S2 और जो अंदर की तरफ है सेल की तरफ will be S3. S3 innermost is sometimes also called as the tertiary cell wall, and tertiary cell wall is chemically and morphologically different from the other layers and is rich in xylins. So I hope that everything related to the cell wall is now clear. Okay. That brings us to the next interesting thing, which is that there are sometimes microscopic channels which traverse the cell walls of plant cells. Meaning कि literally there is going to be a tunnel which will go from one plant cell to another plant cell, and it will literally go through all the layers which we just discussed. It will go through the secondary cell wall, the primary cell wall, the middle lamella, the primary cell wall and the secondary cell wall. And through this channel, it will enable transport and communication between the two cells. Literally, their cytoplasms will extend. So material from here can literally pass through and go here. This connects the cytoplasm of the neighboring cells. And this, dear students, forms a protoplasmic continuation. So the cytoplasm or protoplasm of this cell continues into the cytoplasm or protoplasm of this cell. So this continuation which you see here is called as a symplast. Not important in this chapter, but very important in a few other chapters. So this, dear students, will be called as a symplast. And this whole bridge through which the symplast, the connection is passing, this whole bridge is going to be called as plasmodesmata. It's called as plasmodesmata. Singular, of course, is called plasmodesma, and plural will be called as plasmodesmata. So I hope that this last concept regarding the cell wall is clear with everybody as to what is plasmodesmata. They are going to be protoplasmic connections between the adjacent plant cells. These are going to pass through channels which are through and through the cell walls, and this protoplasmic connection is also called as symplast. The whole structure is called plasmodesmata. So let's see what the NCERT has to say about all of this. So I've already explained all of this, and there is only one paragraph about all of this in NCERT. But we discuss this in so much detail so that in the further future lectures you will be able to easily understand what I will mention in that particular part. So आगे चल के अभी बहुत आसानी होगी अगर ये सब कुछ आज अच्छे से समझ में आया तो. So फिलहाल देखते हैं NCERT में क्या लिखा है. So the NCERT says that, as you may recall, a non-living rigid structure called as the cell wall forms the outer covering of the plasma membrane of fungi and plants. The cell wall gives shape to the cell, protects the cell from mechanical damage, protects the cell from infection, helps in cell-to-cell interaction. So along with that, it also provides a barrier to undesirable macromolecules. So all of this that you see here are functions of the cell wall as given in the NCERT. Algae also have cell wall, but it is made up of cellulose, galactans, mannans and minerals like calcium carbonate. Whereas जो मैंने तुमको सिखाया जो higher plants में, so algae are lower plants, in higher plants you are going to have cellulose, hemicellulose, pectins and proteins. So cellulose हर एक plant में common है, भले ही वो algae जैसा lower plant हो या कोई higher plant जैसे कि angiosperm हो. But in the case of algae, you have galactans, mannans and minerals like calcium carbonate, whereas in higher plants you have hemicellulose, pectins and proteins. The cell wall of a young plant cell does not have secondary cell wall, it only has primary cell wall. The cell wall of a young plant cell, the primary cell wall, is capable of growth which gradually diminishes as the cell matures. And then उसके ऊपर secondary cell wall deposit होता है from the inner side in S1, S2 and S3. S3 can also be sometimes called as the tertiary cell wall, which is rich in xylins. The middle lamella is the layer mainly of calcium pectate, which holds or glues the neighboring cells together. The cell wall and middle lamella of adjacent cells may be traversed by a connection of cytoplasm called as the plasmodesmata. So I hope that this entire paragraph, which is the only thing that they have given in NCERT about the cell wall of a eukaryotic cell, is now absolutely clear to you. Great. So this picture illustrates for your better understanding the various plasmodesmatal connections. So if you've understood everything so far, this picture should be absolutely crystal clear to you. So these yellow cytoplasm wale cells, you can notice here, they have a purple nuclei. This large thing is a vacuole. The green part is, the dark green part is the cell wall, and this light green part is going to be the middle lamella, and these cytoplasmic connections are passing through here. They are called symplast, and this entire structure through which they are passing is going to be called plasmodesmata. So this is all the plant cells together showing the various plasmodesmatal connections. Now, just to finally finish this entire topic of cell wall, I want you to have a look at this diagram. This diagram shows this structure here, brownish structure, as the middle lamella, and these are all the cellulose, hemicellulose, pectin fibers which are forming the primary cell wall, and all of this is secreted via this plasma membrane, and here is the cytoplasm of the cell which is going to secrete all of this here. Now, as the cell grows, this will be pushed towards one side, and from this side, there will be deposition of more cellulose and more lignin and other such compounds. So this is what is going to happen. The cellulose fibers which were there will become thicker, and they are going to be joined by lignin, and there is going to be much less space or much less matrix within it. So you will notice कि ये जो अभी ऊपर दिख रहा है वो है तुम्हारा middle lamella. ये जो तुमको यहाँ पे दिख रहा है, ये this structure which you see here is the primary cell wall. ये पूरा primary cell wall इधर क्रश ही हो गया, और ये जो तुमको यहाँ पे दिख रहा है, this is the secondary cell wall. So I hope that this structure now is clear to all of you. And with this we finish everything related to the cell wall in extreme detail. So now, dear students, we come to the last part of eukaryotic cell, and that is something which all of you now know absolutely clearly. So now it's just going to take 5 more minutes to discuss the difference between a plant cell and a animal cell. So what are the differences that you already know? A plant cell is usually going to be larger, and an animal cell is usually going to be smaller. A plant cell is going to have a cell wall. An animal cell does not have a cell wall. A plant cell will not be able to change its shape. An animal cell will be able to change its shape. Plant cells are autotrophic. They can prepare their own food. Animal cells are heterotrophic. Plant cells are autotrophic because they have chloroplast, so they can produce their own food. So they have chloroplast, whereas these animal cells do not have plastids. The storage material in plants, the carbohydrate which the plant stores, is starch, whereas the carbohydrate which an animal cell will store is going to be glycogen. So the thing which gets stored in a plant cell is starch, and in an animal cell it is going to be glycogen. Plant cells have this large structure in the center called as vacuoles, which we have already done. So plant cells have a large vacuole, whereas animal cells do not have any such vacuoles. Because of this, in the plant cell, the nucleus gets pushed to one side. As you can see in this diagram. So here the nucleus is peripheral towards the periphery, whereas here the nucleus is more or less in the center only. In the case of a plant cell, you are going to have no lysosomes, and you are not going to have any centrioles or centrosomes. But in the case of an animal cell, you are going to have lysosomes, and you are also going to have centrosomes. So you may not have noticed this, but we have finished 10 differences between a plant cell and an animal cell. 10 differences. Let's see what they are. Usually larger in size, usually smaller in size. Have cell wall, do not have cell wall. Cannot change shape, rigid. Can change shape, they are flexible. They are autotrophic. Animal cells are heterotrophic. Plant cells have plastids, chloroplast. Animal cells do not have any chloroplast or plastids. Plant cells, the reserve food is in the form of starch. In the case of animal cells, it is in the form of glycogen. Plant cells have a large central vacuole. Animal cells do not have a vacuole. Because of the large central vacuole, the nucleus is peripheral in the case of a plant cell, and it is central in the case of an animal cell. In the case of a plant cell, lysosomes are rarely present, whereas in an animal cell, lysosomes are mostly present. Centrosomes or centrioles are absent in a plant cell, whereas they are present in an animal cell. So this, dear students, is the difference between a plant cell and an animal cell. The difference between the two main types of eukaryotic cells, plant cells and animal cells. I would want you to just take a screenshot of this right now so that you can use it for future reference. So that, dear students, finishes off the entire eukaryotic cell. Everything about the eukaryotic cell is now over. We have finished the nucleus, we have finished the living components or the organelles, we have finished the plasma membrane, and as of today, we have also finished the cell wall. So when we meet for the next lecture, we shall be completing, or rather we shall be starting with the prokaryotic cell. So dear students, I hope that you've understood everything that we have done today. This concludes today's lecture. I cannot wait to see your smiling faces in the next lecture. Please work hard, be nice. Thank you.